because today is a big day. We're going to try to wrap up kind of what we know. We've covered a long, long road, right? Since the humble beginnings. And since we've kind of covered this various ground of the embryophytes and the evolution of the land plants, we've learned quite a bit. We've learned quite a bit about form, function, underlying physiology, as well as phylogenetic relationships, how we can make sense of the diversity of plants on our wonderful planet. So we're going to try to put all this together. And this is actually going to be a pretty critical lecture because I'm going to give you a few tools that will help you put all this information together. And what I'm going to cover today is going to be essential because it's going to be a core component on the final exam. And on the final exam, we're going to ask you to write an essay to try to pull everything that we've learned this semester together. And what I'm going to show you next are a few of the kind of key tools that I would like you to use to try to synthesize all of the information that we've learned this semester, okay? And to start, I want you to think about what plants are thinking. Now, obviously they don't have brains, they don't have neurons, plants don't think, but if they could, in air quotes, think, what would they be actually thinking? So if you could read the mind of the plants, you would hear things like, if I could just put on some leaves with a lower specific leaf area, remember that's the area of a leaf divided by its mass, it's a measure of allocation. So if I could just put on some leaves with a lower SLA, then I could send my future kids to college and have some more time to respire. Or maybe you'd hear things like, so here I sit surrounded by my neighbors, not knowing what the future is going to hold in terms of climate, maybe hotter, maybe drier. So how much should I spend on roots this year? I wonder if it's going to rain a lot. Or maybe you'd hear things like, gosh, you know, last year was pretty crappy. I'm just sitting here surrounded by my neighbors, and I had a pretty low photosynthetic rate last year, and I lost a little bit too much water. Plus, all my friends died. I wonder if investing in some low density tissue, indicating maybe lower carbon allocation, would actually increase my chances to feed my future kids. These are actual quotes that plants are thinking. Well, obviously they're not thinking, but this is what natural selection figures out. Okay, so they don't think, but how and do they make these decisions? Okay, so the answer is natural selection. And I want you to think about the answers to these questions that plants are thinking in terms of plant life histories and in terms of plant fitness, okay? So we're gonna have to understand what we mean when we talk about life histories and what we mean when we talk about fitness, okay? So in terms of plant life histories, I want you to think about differences in life history as different strategies than that species have to deal then with a highly variable world, okay? So plant life histories are a collection of phenotypic traits that impact the fitness then of individuals via their effects on growth, development, survivorship, and fecundity. Fecundity, remember, is reproduction. All right, so plant life histories are these collection of traits, right, that impact fitness. All right, so what is fitness? So remember, fitness is a relative measure. It's a measure of the reproductive success of a phenotype relative to other competing phenotypes. And it's a measure of both survivorship and reproduction. It's a measure of the ability then of a phenotype to both survive and reproduce, okay? So this is quite important because I'm going to ask you to wrap up everything in terms of plant life histories and fitness. All right, so let's think about plant life histories. What are some examples of typical plant life histories? Variation in growth rate, variation in metabolic rate, variation in how long you live. Some plants live a short lifespan, some live a very long lifespan. Differences in survivorship, your probability of death. Differences in, say, the sex ratio of a population, how many boys and girls and so on. Differences in the age of first reproduction and the type of reproduction. What type of reproduction is actually occurring? These are typical life history attributes, okay? Growth rate, metabolic rate, lifespan, survivorship. All right, now I'm going to give you another equation. I think this is the only equation I'm going to give you today. This is a simple equation because I've given you a verbal definition of fitness. Now we're going to mathematize it. And I'm going to give you one mathematized definition of fitness. We call it R naught. Okay, we may have, I think if you remember back in our early kind of discussions about exponential growth, right? Remember Malthus, right? We talked about R naught because that often comes up, especially when we talk about exponential growth of populations, talking about COVID-19, you often hear discussion about R naught. R naught we can use as a measure of fitness. So R naught is the lifetime reproductive rate of a phenotype. Okay. And I want you to take everything that we've learned about differences in traits and how differences in the environment then influence the form function of plants, and then recast that in terms of thinking about fitness as defined by R naught. R naught is the summation of LX times MX. So what is LX? LX is the probability that a phenotype will survive to age X. MX is the fecundity then of age X, right? It's the fecundity of a given phenotype at a given age. Now notice we multiply the probability of surviving by the probability of how fecund you are at a given age, and we're going to sum then those probabilities across the entire lifespan then of a phenotype, okay? That is going to give us a measure of R naught. And we're going to use that then as our measure of fitness. So as always with fitness, the higher, the better. And remember, always with fitness, it's relative to another phenotype. Okay, R naught. Now, how do you calculate R naught? I'm going to give you a simple way of doing this. This is what demographers do, right? If you work on, you could go in, if you study demography, you could go into working with, say, an insurance company, an actuary, right? And they want to know what your probability of living and dying is at different ages. And they sell you life insurance based on this. Well, as biologists, we use similar things, but we measure demographic life histories and life tables. And so this is a life table, and it's really easy. All I'm showing you here highlighted then in red is this is different ages, okay? We're going to group then all of our plants based on their age. So these are our seedlings, what we call age 0. These are the new plants, our 1-year-olds, our 2-year-olds, our 3-year-olds. And we go out and then do some counting and numerating. And each of these different age classes, our seedlings, 1-year-olds, 2-year-olds, 3-year-olds, we count the total number of individuals then that we see in the population. So notice there's 100 seedlings, there's only 10 1-year-olds, and there's only 2 3-year-olds. Oh my goodness, the old ones are rare. Now, what you do when you calculate a life table, you follow then through the transitions from one life history stage, one demographic stage then to another. And we calculate then the probability of surviving then to the next step. So notice as we go from seedlings to 1-year-olds, your probability of then getting to increasingly older and older age decreases, because notice there's a lot of decrease in the number of individuals, meaning there's increases in mortality. But then what we also do is we keep track of who's actually doing the reproducing. That is, how many then offspring or how many then seedlings then you're producing to the next generation. So if you notice as we go from seedlings, they're not reproducing, but 1-year-olds, there's 5 then seedlings produced. 2-year-olds, they produce 15 seedlings, okay? Kind of prime reproductive years. And the 3-year-olds, man, they're getting a little bit older. They only produce 10 seedlings in this population. Okay, notice what we do in terms of calculating R naught. We multiply LX times MX. LX times MX gives us then our product. And then we sum all of these together to get our value of R naught. Notice it's 1.6 for this population. And that's important because that tells us something about population growth, and it is a measure of fitness because we can compare 2 different life tables from 2 different phenotypes and compare which one has the highest R naught, that is, which one has the highest fitness. Okay, introduction to demography and R naught. All right, so as you then go through each one of these, remember, we use these numbers ultimately to calculate R naught. All right, so we can define fitness in terms of R naught, the net reproductive rate. So in this case, 1.6, that was our value of R naught that we calculated from this population. That literally means that on average, a female in the population, an average female in the population is producing 1.6 females in their lifetime, okay? So one female is not only reproducing, kind of making sure that herself is carried over into the next population, but we have an increase. That is, we have 1.6 more females on average in the next population. The population is growing. So notice if R naught is 1, that means the population is neither increasing or decreasing, but is exactly replacing itself. Every female is replacing herself with another female in the next generation. And so this indicates then population stability. R naught of 1 is population stability. Now, if R naught is less than 1, that indicates a decrease then in the population. If R naught is greater than 1, that is indicating an increase in the population size. And again, remember fitness, you're comparing two different phenotypes or two different populations, and we can compare who has the relatively higher fitness. Okay. So if the net reproductive rate is as high as maybe 2, that means the population then is going to double in each generation. So now we want to then kind of place everything that we've learned, and we're going to start to think about differences in form and function in different environments in terms of how that may impact fitness or R naught. So compare then R naught of two different cohorts or two different populations, right? Again, the population with the greater value of R naught is always going to have the higher fitness. All right, now we're going to use this to then ask, what does natural selection operate on? All right, so again, one measure of fitness, R naught, is the net lifetime reproductive rate. Now, early on in class, we introduced the intrinsic rate of increase, right? Little r. Little r is related to R naught, okay, by this equation. Little r is the natural log of the intrinsic rate of increase, R naught, divided then by t, where t is the generation time of then that phenotype. So remember, some phenotypes live short lifespans, others live long lifespans. So we can relate little r and R naught kind of together. Okay. So what we're going to then do as we move into the final exam is I'm going to ask you questions such as, what plant traits then influence survivorship and fecundity maybe in different environments? As we move from hotter and drier environments or to colder environments or wetter environments, what plant traits then would then be selected? And when we say what plant traits would be selected, that is, which ones then would lead to higher fitness? So higher fitness is, remember, a function of LX and MX, survivorship and fecundity, okay? All right, so what constrains life history evolution? Why can't selection then keep increasing R naught? Why do we not see just kind of runaway values of R naught, right? Well, that's kind of a rhetorical question, and we're gonna get back to that in a second. But we're gonna now start to organize our life histories and kind of everything that we've learned. Now, this is my very crappy plant that I've drawn, my cartoon plant. We have our different organs, leaves, stems, and roots, right? Now, let's think in terms of differences in life histories, in particular, allocation. All right, so remember, leaves are performing photosynthesis, right? And so more leaves you have, the higher rate of photosynthesis for the plant, the more carbon you have, right? The more production then that you can have, the more carbon that you can have to invest in terms of your structural mass, your seeds, right? You can allocate more to yourself, right? More to these costly secondary chemicals, right? So you can increase your survivorship, and also you could increase possibly your fecundity. So obviously, if you have more leaves, higher photosynthetic rates, you could possibly allocate to increase your survivorship and fecundity, right? So we can take our leaves and our photosynthesis and our carbon assimilation, and we can allocate that to resources then that are finite, that we're going to then use to produce maybe fruit or seeds, okay, increase our fecundity, right? Flowers, fruits, and seeds. Or we could take that total production and invest it back into ourselves, right? To maybe live for another day, right? Live for a good time where we can kind of survive and then also reproduce, all right? But now when we think about growing or surviving or allocating them to ourselves, we can allocate it to leaves, or we can allocate it to stems, or we could allocate it to roots, right? These are decisions, right? So we have a bunch of carbon in the carbon bank, and we can spend it on different components of the plant body. So then the goal here by natural selection, the goal of natural selection is then for a given environment, a specific environment, natural selection is then going to balance these investments in order to maximize fitness. All right, so I'm going to say that again. For a given environment, natural selection is then going to balance all these different allocations, allocation to reproduction, allocation to our plant stem and kind of growth, in order then to maximize fitness. All right, so functional traits, these different traits, differences in physiology, form, and function, they ultimately influence differences in survivorship and reproduction, right? So allocation then is key. How are we going to allocate then our photosynthate in order then to either reproduce or grow? So let's think of some examples of how allocation can influence carbon assimilation, right? In different environments, natural selection then for differences in life history, again, defined then by differences in allocation and traits, are going to combine to influence how well a given phenotype is going to survive and or reproduce. Okay. These are strategies, right? So natural selection then, depending on the environment, is then going to come up with different strategies then to deal then with that environment. And it's only those strategies then that can persist, okay, into the next generation. Those are the strategies that we see today. Strategies that fail are no longer with us, right? We don't see the ones then that fail because they go extinct. And the winners then, the winning strategy is then passed on. All right, so we covered many different traits in class, right? So we talked about secondary chemicals, we talked about differences in photorespiration, we talked about roots and how plants can allocate differently to roots. We talked about leaf pubescence, we talked about photosynthesis, right? Differences in plant size. Now, all of these traits together, these different functions, right? They're going to influence how well a phenotype survives and or reproduces in a given environment. So I could ask you, for example, to speculate how these different traits in a given environment may influence survivorship or how they may influence reproduction. All right, now, there's kind of three traits that capture a lot of the variability in plant life histories. The specific leaf area, remember that's the area of the leaf divided by a mass of the leaf. The area of the leaf divided by mass of the leaf. It's a measure of allocation then to leaves, a measure of allocation to leaf area per a given amount of carbon investment. Differences in plant size, differences in seed size. These are kind of key traits that capture a lot of the variability in terms of life history. So differences in specific leaf area influence survivorship in a given environment. Differences in plant height also influence survivorship, but also reproduction. Differences in seed size can influence then your fecundity and your survivorship then of seeds, that is your reproductive success. All right, so trade-offs. We often talk about trade-offs in terms of talking about life histories. A trade-off is a balancing of factors, of which some of which are attainable at a given time or not attainable at a given time. And so a trade-off, you have an indication of what a trade-off is. It's giving up one thing in sacrifice for another, right? If you want one thing, you have to often give up something else. So why do we see these trade-offs in biology? Well, these trade-offs again reflect these allocation principles, right? So for example, we can think about allocation to defense, secondary chemistry. And so this is one of the dilemmas of plants, right? Because remember, the world is filled of insects and herbivores that want to basically eat you down to a nub. And so a fundamental dilemma of plants is allocation to defense, right? But that's going to be diverting resources away from production of new leaf area, right? So secondary chemistry is expensive in terms of carbon and energy. And that's carbon and energy that could be devoted to growing more leaf area, right? So allocation of defense diverts then from growth rate, as well as from maintenance, storage, and reproduction. So this is a dilemma. Do I grow or do I invest more in defense? Right? So what's the cost of defense? Well, let's look at some primary compounds in terms of how much the cost is. That is, how much glucose it costs, sugar it costs per gram then of this compound. Basically, how much it's going to cost me to make a carbohydrate, organic acid, a lipid, a nucleotide, or amino acid, right? So in general, for amino acid, they tend to be a little bit more expensive. I need to make 2 grams of sugar from photosynthesis in order to make 1 gram of amino acid, right? Carbohydrates, a little cheaper. Organic acids, definitely cheaper. But now let's look at secondary compounds. Look at this. For a terpenoid, if I want to make a terpenoid, it costs me 3 grams of glucose per 1 gram of secondary compound. All right, that's expensive, 3 to 1, right? Phenolics, alkaloids, other nitrogen kind of compounds, those are secondary compounds. You're talking about 2 to 1 or 3 to 1 in terms of cost, okay? So pretty expensive. So secondary compounds cost. And it's this cost then that's going to be a loss then ultimately to fitness, right? So allocation to defense or allocation to growth. I could be putting this glucose into growing instead of secondary compounds, but that comes with the risk of being eaten, right? In terms of increased mortality. So allocation to reproduction, allocation to biomass or allocation to reproduction. How much do we allocate to reproduction? Flowers and seeds. How many seeds to make? How much to spend on the quality of seeds? Okay, so we often talk about reproductive allocation versus biomass allocation. Reproductive allocation is the net resource investment in reproduction. That is the fraction of resources that's diverted away from vegetation investment versus reproduction. A trade-off, right? So natural selection then solves this dilemma depending on the local environment. The local environment is going to select for an allocation strategy that does best. Okay, so focus now on kind of these differences in allocation to growth or reproduction. All right, so seeds, let's just think a little bit about seeds. All right, and so one of my last examples here are going to be seeds. So seed size. So if you kind of go out in the world and start collecting seeds, tropical beaches are awesome because you get all these amazing tropical seeds. There's typical seeds then that kind of wash ashore at kind of typical beaches. And you know, seeds really are care packages, right, from your plant mom. And so think of it as this allocation to the future generation. All right, so seeds are these care packages, you know, from mom. So think of it as big fruit and with all these seeds on the inside, right? So these care packages, right? You can have a big seed, and a big seed is a fortunate offspring because a seed, that seed size, is reflected in this maternal allocation of resources, all these stored starch, carbohydrates, okay, nutrients, okay, to fuel the growth of this seedling as it develops. A small seed is a small care package. Small seed means a less fortunate offspring with a lower probability of surviving, right? Because the size of the seed reflects maternal investment. How many resources, right, your mom is basically giving you to survive into the future. So the smallest known seed is the seed, basically the size of a dust grain, right? You can find these in orchids. This is the, look at how small this thing is, about 1 millimeter. It's actually a little bit less if you kind of exclude this wing-like structure. So it's about 0.1 to about 0.2 millimeters then in diameter. That's a really crappy investment for mom right there. But that's okay because mom invests in a gazillion of these things, and with luck, one of them will survive. Now, if you want to find the biggest seed in the world, you have to go into the Indian Ocean, in particular in the Seychelles. Okay, this is the coco de mer. This is a palm tree. This is the largest known seed. Oh my gosh, look at that maternal investment, right? This is what you can find on the internet. This nice honeymoon couple found the largest known seed in the world. Look at all that stored endosperm, all of that storage for the future. And in any given environment, okay, if you go anywhere in the world, go to these different locations in the world and we plot out the seed mass that we find from the different species there. This is on a log scale. We see this huge variation, 1, 2, 3, 4, 5, 6, 7, 8, on the order of 6 to 8 orders of magnitude variation in differences in seed size, right? Ranging from good care packages, huge care packages to the next generation to very small care packages in the generation. Different life history strategies. And you find this all over the world. So why is this? Right? If you look across the phylogeny of angiosperms, you look at the location of the top 20 divergences in terms of seed size. Oh my goodness, look at all these red dots across the land plant, the angiosperm phylogeny. Within any one clade, pick one of your favorite clades in the angiosperms. Even within that clade, you'll find then taxa with small seeds, taxa with large seeds. So natural selection is resulting in certain environments selecting for small seeds, other environments selecting for big seeds. And even within a given environment, we see small seeds and big seeds all kind of coexisting, right? These are different strategies, right? So very large and very small seeds have evolved multiple times just within the angiosperms. So this is interesting. This is reflecting a trade-off. So the larger the seed you have comes at a cost, a trade-off. And that cost is the number of seeds produced. So if you look at the average seed size produced by a given species, that is reflecting this inverse relationship with the total number of seeds produced. So if you're producing a really big seed, most likely you're not producing very many of them, right? But if you're producing small seeds, you tend to produce a lot of them. So there's an inverse relationship, a trade-off between seed size and seed number. And we see that even within species, okay? So you can find individuals then that produce a little bit bigger seeds, some individuals produce little seeds, smaller seeds, and we get this inverse then relationship then. And that inverse relationship is reflected in terms of how fast the relative growth rate then of those individuals. So then we get this very interesting kind of relationship then with this trade-off between seed quality and quantity and quality and the growth rates then of those seedlings. So differences in allocation is going to be then reflected in differences in allocation to growth rates. So relative seedling growth rate decreases with seed size, but seedlings from large seeds have higher survivorship. Okay, so what I want to emphasize here is that what we see is that differences in allocation are going to then be reflecting, depending on the environment, differences in survivorship and or differences then in allocation to reproduction. So why don't all plants have large seeds and high survivorship and high fecundity? Or why don't all plants just invest to the highest level of defense, right? Well, we don't see that in nature because there tends to be this issue of how natural selection shapes life histories in different environments. Because there is this trade-off ultimately between fecundity and survivorship. So if you actually plot a measure of survivorship, your LX, versus your measure of MX, either within a given species or between species, you tend to get an inverse relationship between LX on the x-axis and MX on the y-axis. So there's this trade-off between fecundity and survival. You can't be highly fecund and have a high survivorship. On the other hand, you can't have a low survivorship, right? And also have a low fecundity. So species with low current fecundity have high survivorships. And species with high current fecundity have low survivorships. These are reflecting these fundamental strategies then that plants and actually animals have, all living things on the planet tend to have. It's this inverse relationship between fecundity and survivorship. So why is this? Why don't we have species then with high survivorship with high fecundity. Well, this relates ultimately to this theory of allocation, because ultimately supply of resources are finite and they have to be distributed amongst growth, maintenance, defense, and reproduction, right? You can't simultaneously allocate to all of them at the same time because resources are finite. Right? There's only so much nitrogen, only so much carbon then you can allocate. And all of that trade-off is then reflected in this inverse relationship between mx and lx. So resources are allocated among these competing functions, and that generates then these trade-offs. Now, sometimes plants can kind of get over this and kind of help out. Sometimes we can see photosynthetic fruits. Sometimes stems are acting as biomechanical support to influence survivorship, right? And sometimes you can have vegetative reproduction. But in the end, it's difficult to get over the fact that resources are finite in the world. Okay, and so the diversity of plants are organized along these trade-off axes. Plants can be good colonizers of an environment, or they can be bad colonizers, but be really good at competitors. So you can trade off colonization with competition. You can trade off between herbivory and competition. You can allocate to either more roots versus more shoots. You can allocate to being a highly dormant seed, right? Having your seeds stick around in the environments for a while versus allocating to increased dispersal abilities. You can allocate to living a longer time versus allocating to growth. You can either allocate to survive more, but that comes at a cost to reproducing. You can allocate more to defense at the expense of growth rate and vice versa, and so on. These are several trade-off axes. But what's really interesting is that differences in the environment will then select for differences in survivorship and or reproduction. So how can we organize all these different life histories? Well, take an ecology class, take a life histories class in order to try to make sense of these different strategies than in the real world. So what I would like to end with is how we kind of sort through and talk about the evolution of plant life histories. And this last part is going to be kind of important as well. This is the last important point that I want to emphasize today, because in your essay on the final exam, I'm going to ask you to organize plant life histories and everything that you've kind of learned within this framework. And that gets back to what we learned initially in terms of fitness landscapes. And I'm going to ask you to think about the evolution of plant life histories on fitness landscapes. This is going to be an open-ended question, and I want you to kind of wrestle and kind of grapple then with everything that you've learned in class and try to put it all together. And so if you remember fitness landscapes from Sewell Wright, fitness landscapes are a very powerful tool, kind of like a really good analogy for enabling us to think about how natural selection works, and enables us to make these then links between natural selection and then the diversity of phenotypes that we see in the landscape. So the fitness landscape is mapping fitness values of all possible genotypic combinations of a population, right? And then we can see these darker areas. These are these peaks then of high fitness. And then these lighter areas, these are these troughs of lower fitness, depending on the genetic recombinations, but then also the resulting then phenotype. So remember, genotypes are going to be mapping onto phenotypes. And so natural selection is going to be eliminating these least fit genotypes and phenotypes in the valleys. Think of these different strategies, these different combinations of how you allocate, right? Those are going to be eliminated in certain environments, but certain environments will only favor certain phenotypes. And again, evolution is always going to push these populations up adaptive peaks. So remember back on some of our earlier lectures about this, because I'm going to ask you to come back to those earlier lectures and put all this together. Okay, so the evolution of plant life histories, right? Fitness landscapes are a powerful metaphor, okay? And we're going to then be using then fitness landscapes to try to address these problems. This problem of defining not only possible phenotypes and genotypes, but understanding these fitness trade-offs and how variation in fitness landscapes in space and time in different environments can then select for differences in plant form and function. And then together, those different strategies give us the diversity of form and function. All right, so natural selection is going to act on optimal life histories that are going to maximize fitness in the face of many different trade-offs in the environment. So I'm going to say that one more time. Natural selection in any given environment is going to act on optimal life histories to maximize fitness in the face of many trade-offs. Okay, so in our reading, we've been reading some of Carl Nicholas's book, right? And so Carl is a very wonderful, pleasant, gentle man, and he has a wonderful way of kind of helping to kind of convey and teach kind of how we can think of differences in plant form and function in terms of fitness landscapes and the evolution of plant diversity. And so Carl was actually one of the original innovators of the use of computer simulations, actually in botany, as well as in evolutionary biology. And so when the first computers came out, he realized that we could use computers to help us think and simulate our understanding of the evolution of diversity. And so he came up with the creative ways to think of how, if we go back to the Devonian, back to the Silurian, we think of Cooksonia and the branching patterns of Cooksonia, to then start thinking of how this little dinky little plant, this little Cooksonia with this simple dichotomous branching, then led to this enormous diversity of plants that we see today. And so if you remember back, as we go back to the Silurian, back into the Devonian, we can kind of map out these differences in macroevolutionary time, these major adaptations of these land plants to basically living in the air, right? Tracheids, stomates, right? Differences in branching architecture. Secondary growth, we have xylem and phloem, we have megaphylls, we have seeds, right? All of these are adaptations then to living then in this terrestrial environment, right? So remember this early land plant morphology where we had Cooksonia with this dichotomous branching, we had sporangia then at the tips. Remember sporangia? Remember all of this? And so we're going to use then this fitness landscape and Cooksonia to try to put all of this together. So I don't expect you to memorize this at all. This looks complicated, but it's actually really easy. But what Carl did in his early use of computers in the simulation then of biology was he said, okay, let's take early Cooksonia and then ask what could natural selection operate on in this little dinky dichotomous branching plant with sporangia on top. Well, he said that, well, take this basic branching architecture and you could think about the angles of branching. You could talk about the rotation of then the branching. You could then think about the branching probability as you go from one mother branch to daughter branches. And those are basically the traits that natural selection could operate on in these different environments. And what he asked was, well, let's think about this phenotypic space. Let's think about these fitness landscapes, right? All the possibilities that natural selection could operate on in terms of the frequency of branching, the rotation angle, the bifurcation angle, right? So don't get too hung up on this. All we're focusing on is natural selection can just operate on a few things because this is a very simple initial plant. But this is the space of phenotypic possibilities, right? And so let's think of life on land, differences in light interception, right? Differences in biomechanics, mechanical stability, right? So different environments then will have differences in wind and gravity, but there's also reproduction, right? And so maybe the taller you are, the bigger probability you have of getting kind of picked up then by wind and blown around, right? To increase your reproductive success of finding a successful kind of new site. Water loss, right? Your total surface area is going to influence how much you desiccate in a given environment. So let's kind of define our initial components of maybe different environments we'll select for differences of fitness in terms of how well you intercept light, how well you're able to kind of be biomechanically stable, how well you reproduce, and whether or not you're going to be desiccating or not. So now let's think in terms of R0, fitness, differences in survivorship and reproduction, right? So drier environments then will select for minimizing water loss. Maybe different environments, highly competitive environments, will then select for increase in your reproductive ability or increases in light interception. Right? So different environments then are going to be shaping fitness values or R0 in these different environments. And the phenotypes with the highest fitness will win and persist. So we'll start with our initial conditions, our Cooksonia-like. This is our initial condition that we're going to seed the simulation of evolution with in Carl's early computers. And that's what he did, is that he used the computers then in these different environments to search all these different phenotypic spaces. And then in each of these different phenotypic spaces, he then said, okay, how does differences in water availability or light availability then influence which phenotypes I should expect to see? So he then ran these simulations on these early Cooksonia and let these kind of computer plants kind of wander around then environments that had differences in light availability or differences then in the ability to disperse or differences in the mechanical stability. And then he asked in different environments which plant architecture then best matched your best ability to disperse, your best ability to intercept light, and your best ability to be biomechanically stable. And from that initial Cooksonia design, popped out, this was the phenotype that did best in reproducing because you had all these tips here at the top that could easily disperse. Instead of in another environment, high light environment, these then were the phenotypes that maximized the interception of light the best. From your simple Cooksonia beginning, if you wanted a phenotype that did best at intercepting light, this is the phenotype that you would have. But if you wanted a phenotype that was best at being biomechanically stable, these were the phenotypes then that emerged. So notice, from a simple, humble beginning of Cooksonia, we could then select in different environments differences in architectures that did best in dispersal, light interception, or biomechanical stability. So you could then say, well, what if natural selection, okay, specialized then for one task, such as spore dispersal or light interception, or minimizing surface area to minimize desiccation, or maximizing mechanical stability. From such simple beginnings, you could then have selection lead to these different phenotypes. So these are then adaptive peaks on our fitness landscape that specialize in one task. These are specialist morphologies. But natural selection could also maybe select for two tasks, right? Maybe as in the case of C, okay, these then, okay, are phenotypes that do good for maybe two of those different tasks together, okay? So if we start then selecting for more than one task and then ask what then are those phenotypes then that do best for those two tasks, we start to get a little bit more of diversity in terms of these differing tasks. These are adaptive peaks if you let natural selection then try to maximize three different tasks, okay? So again, M, L, and D in this case is M, maximum mechanical stability, L then being maximum light interception, and D being maximum spore dispersal. These are phenotypes that do really well at three different tasks, okay? In this other side here, if we just go in terms of panel D, L, D, and S, these are phenotypes that do really well at light interception, maximum spore dispersal, and maximum surface area, okay? So depending on how well we select on these differing components, be it one, two, or three of these different components together, we get these differing diversities then of phenotypes. In then this simulated fitness landscape. Here are, if we select then on phenotypes that do best on all of these together. And so if you notice, the more different tasks then that we select on, notice a lot of these different phenotypes start to look like things that we would encounter in our everyday life, okay, in terms of real plants, right? So notice what happens when we select for more of these tasks then, or optima. We get these really highly variable morphologies. And so land plant evolution has really been, think of land plant evolution as exploring this phenotypic space, right? Due to differing selective pressures to either maximize fitness in these differing environments. And so we've really kind of covered this really large ground here. And you can kind of map on to, trimerophytes and the rhinophytes and the zostrophyllophytes, right? And overall conifers and angiosperms, right? These highly variable morphologies are reflecting these differences in different environments for these differing selective pressures. So in terms then of fitness landscapes, if we then kind of start off with our initial land plants, and we then kind of lead to the kind of modern day where we have this incredible diversity that of bryophytes into the angiosperms as well, we have more of these phenotypic peaks because these different environments have led to unique solutions in terms of morphological and phenotypic space. Okay. But in general, what tends to happen is that we have many more fitness kind of peaks, but they tend to be relatively smaller because we're selecting for these many more kind of optimum. Okay. So back to the biology. How then do organismal lineages then search this space? So remember, mutation, recombination, polyploidy, critically important for plants. Hybridization, critically important for plants. Plasticity, plants are actually quite plastic in terms of how they can kind of, within then their ontogeny, kind of change then their traits. So what is going to be limiting then the search? Things like developmental limits, genetic impediments, right? It's natural selection, drift. All right. Now, we haven't had a lot of time to cover kind of what limits then these searches in phenotypic space. Take an evolution class, take a genetics class. Okay, but so the red areas here, these are things that maybe you haven't learned yet. Okay, but if you want to continue to learn, take a genetics class, in particular evolution class will help fill in some of these. Okay, so we've learned many different ways that plants then actually search then the space. All right, so to kind of end up here, so I want you to consider the following. So what are then the ecological and evolutionary ramifications of these very complicated fitness landscapes in the context of the large number of physiological and life history trade-offs? So these are the major adaptations to living in the land, in the air, the synapomorphies, these key synapomorphies then that we've talked about. So remember, we've talked about these different clades. These clades have different key synapomorphies then that define them. And these clades with these synapomorphies then enabled them then to colonize and to persist in certain unique environments. So then to summarize, the more complex selection domains that we have, the more trade-offs we have, right, as land plant evolution then has proceeded, the more adaptive peaks we've had, but also the lower than these adaptive peaks. Okay, so then the fitness differences then between any one phenotype are not as great, okay, because these differences in strategies are all good strategies for a given environment. So we can have small seeded plants within a given environment and big seeded plants within a given environment. We can have plants in a given environment that have short lifespans or longer lifespans because in general, there are multiple ways to make a living within a given environment. So the take home messages, the more trade-offs then that we include, that are included in terms of optimizing your fitness, the natural selection is kind of grappling with, the more optimal peaks then there are in the landscape. So remember, there are all these different trade-offs to growth versus longevity, trade-offs to growth to reproduction, right? Trade-offs with growth and then allocation to secondary defense. We have a lot of different trade-offs. And so the more trade-offs we have as land plants then have diversified and we have more of these different synapomorphies and more of these unique traits, the more then ways we can find an optimal solution for a given environment. So there's more ways of making a living. The more trade-offs you have, the more different strategies you have in a given environment, and the more potential for diversity. So think of diversity as strategies, these allocation strategies, these differences in form and function. And again, how does this all relate to the evolutionary process? So remember stabilizing selection, remember diversifying selection. What does this mean about how you think about these different environments, that different diversity? Oh my goodness, this all sounds like the basis of a fantastic essay question that we can ask you. All right, so here are some questions then to end with. You should be able to define fitness now, and you should be able to describe and label then an equation for fitness. You should be able then to tell us some of the important traits covered in class and how they influence fitness. You should be able to tell us an equation for plant relative growth rate in terms of photosynthesis, in terms of how that influences then life history. You should be able to tell us about fitness landscape and how you can take everything that you've learned together and wrap it up. And according to Carl Nicholas, what are the main selective drivers of plant fitness? Well, remember, he simulated potential differences in selection for light interception, minimizing surface area to avoid water loss, right? Differences in reproduction, ballistic dispersal. And so you should be able to describe then how natural selection to maximize fitness can lead to the observed diversity of plants that we have today. Okay, so that was more than enough and more than enough to leave you on. Okay, so